Today I'm actually going to talk more about links, continue from where Julio stopped and actually use one of his old model simulations from yesterday to sort of build up our base and build links between compartments. Couple of reminders that this meeting is being recorded and live streamed. Zoom logistics and yeah. All right. So quick recap of what we're going to do today. So we're going to look at the epithelium model that you covered in 4.2.2. And we're going to use that base for building our compartmental cells again. Then we're going to use the FPP plugin that Julio just showed you, and we would be putting specific external links between compartments. And the reason why we want to do some of this is because we want to build a model of an epithelial layer bending or folding. And we'll walk through step by step. So I have two main simulations, one which we'll cover in detail together, and the other one, it's already been uploaded in the Google Drive, and it should be marked as model 5_1_3 and 4. So those are the simulations that I think it would be best that if you download, and we'll walk over what is being done, but I think it might be too involved for us to cover it during this session. All right. So motivation-wise, why are we interested in epithelial tissue folding? So during processes of development, morphogenesis, you actually have cell shape changes that drive the whole tissue shape. And in these cases, cells actually exert physical forces that can actually change the shape of the tissue. So part of understanding that, because CompuCell provides such a nice way to model cellular behavior, we can combine these cells to form tissues and basically model forces between these cells that allow us to change the shape of the tissue overall. In most of these folding or bending problems, what you'd have is some sort of asymmetry that would indicate where excessive forces have been are being generated, or some sort of asymmetry that would basically change the shape of the tissue from a flat surface to that of a folded one. One of the important players that we know that actually drives tissue shape is the actin-myosin cytoskeletal network. So a popular way to basically drive invagination of an epithelium layer is basically having a high density of myosin motor proteins, which basically generate these contractile forces, help reduce the apical surface area, and that basically generates a folded shape. So this is a model that we would be working in. Other examples of this, Julio has already talked about convergent extension. There are also processes of intercalation that cells could undergo to basically change the shape. All of these are possible to model in CompuCell. But for today, we are going to actually focus on the problem of apical constriction and how we are going to invaginate a tissue using our FPP links. And based on the last session, I think you already have ideas on what we are going to do, because you already know the important parameters of target length and lambda distance. So those are the parameters we'll be playing with to basically generate our shape. So just the objective for the session is that we're going to basically start with an epithelium, which is going to comprise of an apical and basal site that you saw Julio generating yesterday. And what we're going to do is put specific links between the apical compartment, and we're going to change certain properties of the FPP links so that we can invaginate this tissue. So the starting simulation should be marked module 5_1_1 tissue folding. So if you want to, you feel free to also download it and move along with that, or start to edit, and we're going to walk through these steps again. So when Julio generated the epithelium layer code yesterday, he did not add the focal point plasticity. So we're just going to quickly walk over all these steps, add the focal point plasticity, just as a quick recap. All right. So the steps that he adopted yesterday was he set some dimensions for the X and Y lattice. So he set that to 350 and 100 for the X and Y respectively. He set periodic boundary conditions, and he set 4 different cell types of apical, lateral, basal, and ECM. So I'm going to stop here for a moment, let you begin to edit, go through the first steps of setting up the lattice parameters and the cell types. And please let me know when you're done with that so that we can move on. If you feel like it's too simple and it's a repeat, feel free to download the simulation and run it and see what it does. Feel free to play with the parameters of the code and see what changes. I'm going to wait like another 30 seconds. Right. Looks like most of you are done. So after setting up the 4 cell compartments, the 3 cell compartments and the ECM layer that we're generating, we're going to move on, ignore any of the chemical fields, and we are going to add a couple of plugins. So the only plugin that we did not include yesterday was the focal point plasticity. So make sure you have the contact plugin in, the contact internal plugin in, the focal point plasticity, the volume plex, cell neighbors, center of mass. So that's basically our simulation, and we're going to set up some links between the compartments now. Okay, so the first thing, if you remember from Julio, is that he had a way of already generating the layer using uniform initializer that did not require any extra input from the Python scripts. So let's go to line number 255 of our XML file. And in the uniform initializer code, make sure that we basically set up the layers. So what he had done was he had stacked up the ECM. On top of it, he put the basal compartment. On top of that, he put the lateral compartment. And on top of that, the apical compartment. So if you're looking at the Y dimensions, you could look at line number 280 to 282, which basically sets up the ECM dimensions. So you're setting up a width of 35 pixels, and then you're going on to set up 7 pixels for each of these compartments. So from the top to the bottom, you have 0 to 35, then you have 35 to 42, then you have 42 to 49, and 49 to 56. And that basically changing the dimension in the y-axis stacks the compartments on top of each other. And you have in the x dimension, you're basically setting up the entire region of 0 to 350, but you're giving them a width of 7 units so that each of these compartments would have a 7 by 7 unit. Right. So these are all these steps you have done yesterday, just redoing the simulation so that we could look at the FPP link plugin. Right. So I'm sorry. Before we to any of the other steps, one thing that we should do is make sure we have the correct volume for each of the ECM cells. So make sure to set up the target volume appropriately in the volume plugin. That should be in line number 31 to 36. So at this point, what we have in the simulation is all that you had yesterday, except that we added the focal point plasticity plugin. And we have not changed anything in the focal point plasticity plugin at this point. But so if you just run the simulation, let's try that, run the simulation and tell me what you see. So Norma, the slides should be uploaded in the course slides folder. It should be called module 5_1 with the links, epithelial cells, and tissue folding. If not, Hayden, could you also send the share the drive link if that's helpful? So all we did so far was went through the FPP steps. We adjusted the uniform initializer steps so that we created these 4 stacks of cells from the ECM to basal to lateral to apical. And we made sure we had the correct volume adjustment for the ECM cell. And that's all. So were people able to just run the simulation and see what happens? I think Julie already told you the answer in his demo of what you're to expect. But let me know if people are able to run this. I need more time.
Priyom, can you see the polls now?
Yes, I can.
Okay, great.
Okay. All right. All right. So if by itself you don't modify any of the FPP properties and you let the simulation run, automatically FPP creates links between all cell pair types in the simulation. It also, the automatic parameters are also set at max junction of 1, which is what somebody pointed out, that you don't see links between every pair. But automatically FPP will generate all of these links, and it's not going to be specific. So if we want the FPP links to be specific to a particular cell or compartment, we're going to modify properties in the FPP plugins. So here, the idea is that if you just look at any of the FPP snippet, you will see that it has this the syntax, which Julie already went over, but I'll just repeat. So you have the parameters tab that lets you put links between cells. You have the lambda parameter that lets you control the strength of these links. You have the activation energy that gives an extra push for the link to form. You have the target distance, which is the requested distance between any 2 types of cells. You have the max distance over which the links will form, under which the links will form. And you have the max number of junctions, which is the maximum number of links a cell can form. So the 2 parameters for our goal, which is basically to change the properties of an epithelium so that it gets folded, are going to be the lambda parameter and the target distance in this case. So those are the 2 things that we'll be modifying. And because we only need links in our simulation between 2 compartments, only between the apical site, we're going to be eliminating most of the FPP syntax, but keeping only the tabs between apical open. So we could, in principle, implement links between other sorts of compartments. Somebody also asked about the internal parameters tab. So we could, in principle, put links inside the cell and control growth and other properties of the cell itself using FPP links. But for this simulation, we could choose to ignore any other links. But feel free to experiment and keep any of these tabs on to see what those links do. All right. So just to recap what I was saying, so we're going to make a few changes to the FPP focal point plasticity. We're going to remove all other parameter tabs that generate links between any of the other apical lateral compartments, for example, apical basal, any other compartments that does not say apical to apical, we're going to remove, because all we're interested for this is the link, are the links between apical and apical. We will also switch in the tab we will also uncomment line number 88 so that we have the local tab on, because what we want to do is to change specific properties of these links at specific points in time. We need access from our Python scripts. So switching on the local tab will allow us to manipulate changes in the Python script. Right. Gonna look at the poll. All right. And let me quickly check out this stuff. Grayed out in the vision. So Sharon, have you what sort of can you make sure you have an apical to apical tab? Can you make sure that all the parameters are set to non-zero values? Make sure that the focal point plasticity plugin actually has these code snippets that are forming links between them. If the color is an issue, just try maybe changing the color to something else to see if it pops up. But if not, let's touch base soon. Okay. Okay, so there are 3 people who still are working on the exercise. So again, just to recap, what we're doing now is trying to modify the FPP simulation, the FPP simulation plugin region, so that we only set links between apical and apical compartments, and we're going to get rid of everything else. If you want to play around with putting links between different compartments, you can feel free to do so. But for this exercise, we're only putting in links between the apical and apical compartments. Right. So the other thing we're going to do is, because we want the properties of the FPP links to be controlled from inside Python, whatever is in the XML script, we're going to set those to 0 so that we can control it from our steppables file. So we're going to set lambda to 0. We're going to set target distance to 0. We're going to set max distance to 0. We're going to set maximum number of junctions to 0. We can keep the activation energy as it is, but in the new versions of CompuCell, this can also be manipulated from inside the steppable file. So I have kept it at -50 here, but you can also remove it and reset it back in the main steppables file. So if we do this now and you actually play the simulation again, you should be able to see that the links have actually disappeared. So even though you have switched on focal point plasticity, just by setting in the actual parameters that create the links, we're able to make them disappear. And yeah, neighbor order, we can set it at 1 as well. If they're not 0, what happens then? So if you have any of these parameters, for example, if you set a particular lambda, then you will see these links appear. So I have just set the energy function that's associated with this, all of the terms, to 0, so that they don't, they're not created from XML anymore. Yeah, so you could for the first part, you could have some XML values as well, and you could also have some values for Python values as well, Megan. But yeah, we can just start off with the Python values. But yes, it's totally possible to have to set up some initial XML values that you can later on manipulate from the Python file. Right. So the other thing, just to make sure that we have the right contact energies. So if you remember Julia's chart yesterday, he had sort of a hierarchy in which you had the basal compartments prefer the ECM. You had apical and lateral compartments actually not like the ECM at all. You had the medium and apical compartments like each other more than the medium or medium to lateral and much more than the medium to basal. And you had the lateral and lateral compartments, which had a preference for each other. So based on that size chart, we can keep the same energy functions that he had on. And those are the values I'm using right now. For the internal contact energies, we can do the same, where to prevent some sort of unmixing between the compartments, we actually said that the energies between the compartments have to are set to 2. So Michael's asking about the getting rid of the internal parameters. Yes, let's do that for this simulation. If you want to also manipulate the way that the cell shape evolves, you can put internal links to sort of like change and extend or shrink them, and that will like bring the compartments closer, push them apart. So you can put internal links, but for our purposes, we're getting rid of all the internal links. And I'll quickly go back to this slide. This is so if you got rid of everything and made the adjustments, this is the focal point plasticity code that should remain. So basically you'll have about like 83 to 102 lines where I have gotten rid of all the external compartmental energies. I've gotten rid of all the internal compartmental energies. And all I have done is set out some, set the, all the attributes of the focal point plasticity to 0 except the activation energy. All right. Okay. So, sorry. So yeah, this is the step. So this is the final XML step, which we were working with. Some part, it's 1225. It's basically 35 times 35, but 350. Yeah, 35 times 35. Yeah.
No, I meant the lambda volume. Have you made it more rigid than the cells?
Oh yeah, I haven't actually. You can set. You can try changing it to something else. But yes, you can also set it to 0, and
Okay.
yeah, everything will sort of like mix apart. So as long as they're not making contact with the medium, you'll see that they'll actually stay in the simulation. They might appear a bit pixelated. Right. Okay. So, so these are the XML steps. I'm just going to quickly look at if there are any updated polls. Okay. So now we're actually going to move onto the Python steppables file, because what we're going to do is make sure that, so until this point, like Julia pointed out yesterday, that the cells do not know that they're part of the same cluster group. So we have to sort of like organize them. And the way that Julia did it was, he had the epithelial layer set. And before any Monte Carlo steps actually were undertaken, just with the initial base simulation, because each cell was created by stacking of these compartments, each of their X center of mass would have been the same. And that helps because now you can read you can start off with any apical cell, you can read its center of mass, and you can say that, yes, if I can find a lateral cell or a basal cell, which has the same center of mass, that must mean that we are created in the same place and we are the same, we belong to the same general cluster ID. So that's what he did yesterday in the Python steppables file in these lines 15 to 25, I believe, that he modified. And yeah, so doing this will basically ensure that your apical compartment, your lateral compartment, and your basal compartment actually know that they're part of the same cluster ID. Yeah, so the only reason is if you have found the right pair, sorry, the question was, what is the reason for using break in this code? So if you're basically parsing through a huge list and you have found the right pair, then there's no need to like continue any further. So you can remove the break and it'll still work, but it just saves you a few for loops if you have found that right pair. I believe that was the motivation for doing this, Julia. I don't know if you're still online, but that would be my interpretation. Right. Okay. All right. So now what we're going to do is, this is the part where we have to use the cell neighbors. And because we're going to manipulate some of these like Python code, if this is too much all at once, feel free to look back on the uploaded code, which is still the same version of 5.1_1. And you can copy the syntax. But basically what we want to do here is we want to go to line number 27 of our steppables file. And what we want to do is we want to set up links between apical compartments. And how do we do that? So a cell should know that it's what its neighbor is, because it may be next to a lateral cell, or it may be, I mean, hope not the layer inverts, but it could be next to a basal cell. So we need to find out for each apical cell what its neighbors are. Once we have identified its neighbors, we need to extract that whether that cell is an apical cell or any other cell. If it's an apical cell, what we are going to do is we're going to put a link between those two cells. So the code, the first couple of steps are basically going to each of these apical cells, which is that line number 28, that's looping over all the apical cells. Then it's trying to find out what the neighbors of that cell are. If it's a neighbor and the neighbor type is apical, we're going to put an FPP link. Right? So this is the syntax for basically trying to parse through the cell neighbors of any given cell. I'm just going to wait a little bit longer because I don't know if you have seen this before. Also, this syntax is available in CC3D Python, visit, and cell neighbors. And I hope you already know how to loop through all these cell neighbors of a type. So the list of all the cell members by a given type is contained in the list called self.cell list by type, which is what we're using. And giving it the attribute of self.apical are only going to pick up cells that have the apical, that are the type apical. And then the next step is that we have actually gone through and tried to collect its neighbors, and we are trying to pick up which of these neighbors are apical. Hopefully that makes sense. Going to the chat. Oh, that's a great catch. Thank you, Izzy. Yes, absolutely. Absolutely. I missed that in the syntax. That should be, it should be the correct version in the uploaded code. So Izzy pointed out that line number 31 should say if neighbor.type equal to self.apical. You need that self attribute because apical is a word that doesn't mean a thing to CompuCell. So self.apical is a cell type. Thank you. I'm going to actually correct this line in the chat as well. Okay. All right. So if this is okay, what we're going to do next is actually add the focal links themselves. So now, if we covered this part where we parsed through all the apical cell types, we got the neighbor, the neighbor list, and we checked if neighbor type was apical cells. We're now going to go to CC3D Python. We're going to focal point plasticity, and we're going to actually create an FPP link. So CC3D Python, focal point plasticity, create new FPP link, and it's going to generate some syntax for you, which is going to have this name called self.new FPP link. And you will see that it has a few options. So the options are basically it allows you to choose the two cells between which you're going to put those links. In this case, our outer loop at the beginning of line 28, which is this cell that we're going to, that we're trying to find a connection for. We have already gone through the neighbor list and we have found out the neighbor. The next two parameters are basically lambda and the target distance. And the third parameter is the maximum range over which you're actually forming the links. So basically, it's the same thing as you saw in our XML snippet, but Python allows us to basically modify this in the code. So the first parameter is that of the cell. The second parameter is whatever neighbor to which we're forming links. The third parameter is that of lambda, which basically controls the strength of the links. The target distance is what is the required distance that we want to form between them. And then the last argument is basically over what range do you want these links to form. And you would remember that the width of your compartments that you chose was 7. So we are going to set a target distance of 7 for now for this case, and we can set a lambda of 10. Right? So let's pause here for like maybe 30 seconds, make sure we have this code in, and hopefully, can you please let me know that when you run it, you actually are able to see link-like objects, and to monitor the chat as well. So Sarahna, I hope I'm saying that correctly. So it's the maximum range over which the links will form. So if you have a distance of say 200 pixels, and beyond that, if you try to like set this particular link, it's not going to form. If the cells are closer than 200 pixels, then that link is going to form. So it sort of sets the maximum length over which you're actually going to try to find and form that link. So is this simulation working for people? Yes, no, any issues that I can help with? All right. Thanks, Bharat. Anybody else? Okay, perfect. Thanks, Caitlin. Okay, so so this simulation, if you just look at it, all we have done is set up links between each of the apical compartments. And all of them have similar properties, so nothing has really changed. If you let the simulation run for a while, you might see this, some sort of like rearrangement in terms of the cell body itself, but nothing else is going to change from this because everything is uniform. We also have periodic boundary conditions, which means that these cells are actually connected back to each other. So we don't have, so we haven't changed anything in the simulation so far. So and if you look back at the problem that we started off with, which was that we wanted to create some sort of an invagination in the middle of the epithelial layer. So we have to now think about what we would change in the FPP parameters to sort of do that. Sure. So Megan, yes, absolutely FPP links, but what would you, and the surface too, what kind of properties would you change So if I showed you the picture from the last slide, which was just a flat piece of epithelium, what are the properties that could change? Increasing the lambda is an absolute good way. Would you increase the lambda everywhere all at once, or is there something specific you do here? Locally, so Bharat's answer was locally. That makes sense, right? Because if you want to induce some sort of folding at a specific site, indicating that there must have been some local sort of a source of asymmetry in that region, we need to find we need to create a local spot and change the properties of that region. Thanks, Megan. So what we're going to do is exactly what you have thought about, which is we're going to change the properties of this tissue in the middle. And to do this, again, this is where the Python steppables file actually comes in handy, because we can just like you saw it, we can get the FPP links, we can modify their properties. So while the simulation is actually running, we could access the properties, change their lambda volumes, change their target volumes, and create all sorts of specific tissue properties that we want. Okay. So the next step, what we're going to do is the following. So we're going to try to bend the epithelium, and we're going to do it in 2 steps. So I want you to now go to the step function of the steppable class that you had, and go to line number 49, around line number 49, assuming you haven't added anything else. We're going to do 2 things. We're not going to set it immediately. We're going to let the tissue evolve a bit, adjust according to its contact energies and everything. We're going to pick up a specific time point, which is 500 Monte Carlo steps. Then we're going to parse through the apical cell list again. And just like you all guessed that we need to find these local group of cells to sort of behave differently, we're going to use our center of mass plugin to pick up those cells locally within a region. So in this case, we're going to pick them up between, say, 125 and 225 between the center of the simulation. And then we're going to, again, call on our focal point plasticity plugin. Focal point plasticity plugin, and then we're going to adjust the target volume, the target distance of these specific links, and the lambda distance of these specific links to different properties than what we had before. Right? Let me look at the chat. So Megan, it could be between these 2 XCOM. Not sure I got the question. Yes, I mean you could, in principle, put links between any 2 XCOMs. If even if they're separated in space, you could create a link by just associating the 2 cells that you want a link between. Here we are using the center of mass positions to just isolate that group of cells, because we only want to pick those cells to change the link properties of. Okay, so again, what we're doing is going to the code. We are making 3 different sets of changes. We are going to make an topmost outer loop that says that's going to specify the time period at which we're going to put this asymmetry in. Then we're going to loop over all of our apical cell members. And then we're going to only pick up those apical cells, which are between a particular region of space. And we're going to differentiate their link properties from the rest of the cells in the simulation. Right. So in the previous one, where we basically were setting up the initial properties, that's in start. And now these ones that we have are in the step function. And feel free to also look at module 5.1_2 tissue folding for the syntax if they're in confusion. So, and I thank you for pointing that out because it's important to, oh, is it not in the module? I'm sorry, Ajay, is it not in the demos folder? I'll make sure in a second. So let me quickly go back and just recap. So when we set up that FPP link properties, we could, in principle, set those up at any Monte Carlo step. In the simulation, what we did was we set it up at the start function, which was basically the slide. So here, when we set up these functions, which was at line number 28, this was part of our start function. So we basically looped through all the apical cells and we created these FPP links. Now, because we want to make these changes, we want to make these changes at a specific period in time, we now need to go to the step function because the step function takes in the Monte Carlo simulation input from CompuCell. And so it allows us to add any piece of code at any point during the simulation. So that's why the rest of the changes that we're making is at the step function. Right. So Megan's asking about why there is MCS. No, it's not. I mean, if you're not familiar with Python, that sounds very odd, right? So the way that Python's if loops work is when you try to sort of like compare two things, whether one thing is equal to other, the syntax are that it takes either two equals for equal and exclamation equal for not. So basically, if you want to check if, for example, if 2 equal to 3, you do this. Or, if you wanted to check if 2 is not equal to 3, and that should give you an output, you do something like this. So that's basically Python syntax for checking if two quantities are equal. So the step function, one of the attributes of the step function are these MCS values, right? Which is the Monte Carlo step. So what it's doing in the step function is taking in this parameter of Monte Carlo step, checking what that value is. If it's equal to 500 Monte Carlo steps, it's going to make this next level of changes. Hopefully that helps. Okay, so I'm going to quickly check the poll. Let me check if, all right. Right. So looks like there are 2 people who are not who need more time. I can wait another like 30 seconds, but if you have any more pressing concerns with CompuCell itself, please reach out to either Hayden or James, and we can probably help you during the duration of this session. All right. So once you set this up, I don't know if people already tried running the simulation. Sorry. So once you set this up, why don't we go ahead and run the simulation, and maybe you can tell me what you see. So for people who are done with the exercises, are you able to run the simulation? Do you see something crazy happening after 500 Monte Carlo steps? Okay. I like the word modus folding. You could, Michael, you could go even crazier and set the target distance parameter to higher than the value we have right now if you would like. Also, wait for a couple more Monte Carlo steps. Okay, nice. So so it looks like you're able to get some sort of folding in the tissue. And all we did was in the lines 56 to 58, what we changed were the target distances and the lambda distances. So remember, initially we had all of the lambda distance to be the same at 10, and we had all of the target distances at the same at 7. So now shortening out both the target distance and increasing the lambda distance in that region allowed us to sort of create this asymmetry that we have been talking about. And so you sort of get that folding. Now, if you're not happy with the amount of folding that you get, you can move on and sort of try this. You can either try changing lambda distance to a slightly lower value and see what you see with the simulation. You can also change target distance to 2, and you can tell me what you see. One more change that you can try making in the CompuCell code itself is sort of commenting out that line that says pick up XCOM between 125 to 122, and basically allowing everything to sort of contract out at once. And maybe you can tell me what you see in that case. But because this is the main exercise, I want to make sure that everybody is able to get folding and can tell me if you're able to change the amount of folding by changing the values of these two parameters. Sure. So in this case, what you're, so Lily's asking if, why does it arc down instead of up? So what you're essentially doing is shrinking that epithelial region. So what you're trying to do, force the apical compartments to do are sort of like come closer to each other. And when they come closer to each other, the cells which are on the absolute border, which do not feel the force from the cells from the right, but feel the force from the cells from the left, are going to sort of get pulled towards the left. And then the same thing is going to happen to the cells from the other side, basically cells which are pulled to the right, but not to the left. And when that combination happens, essentially you're going to start getting an arc. You could actually try at the same time, if you had other links of, for example, the basal links, you could try either shrinking them or trying to actually extend them and see if you see anything suspicious or funny combined with that. So if you want to add, so in this case, like I pointed out again, you only have links between the apical compartments. You can also add links between the basal compartments or lateral compartments or any of those. You just need to make sure that they are, they have the parameter tags in the XML script, and you can modify them in the XML file or inside Python using the FPP focal plasticity links. Okay. All right. So are people able to get the epithelium correctly? quickly. So I think we have like 20 minutes or so. If you have been able to get the epithelium to bend, are there anything interesting that you have been able to spot with the changing the parameters, uncommenting maybe the specific region in which you're activating the links, or even expanding the region? For example, you can set cell, you can pick up, instead of picking up cells between 125 and 225, you could actually extend the whole simulation range and see what it does, or even reduce it more than this. Okay. Okay. So what I thought I could do next was, now that we know how to change up FBP properties locally, we could play around with another simulation that would more closely talk about a biological process called segmentation. And I mean, the arching goal would still be to explore more FBP properties by trying to break them, or trying to manipulate them in Python. And I thought we could go over the code together and see at the end if you would like to add plots and visualization techniques to sort of like play around with the code, right? Is this a good time to move on, or do you need more time? Right, I'm going to move on then. Okay, so now what we're going to talk about is sort of the using FBP links to mimic epithelial tissue breaking during something like somatogenesis. So far what we have done with FBP links is we have sort of simultaneously activated all the links of a particular region. And we have sort of obtained a shape that folds and gives the tissue an invaginated shape. But what we could also do is create our whole epithelium, and we could periodically sort of break it into chunks. So does anybody have any ideas on how we could achieve this, and what we could sort of like change in the original code? So in the original code, what we had was we had an epithelium to begin with, and then we specifically changed the properties of the cells in the middle, and we sort of got them to invaginate. Suppose now we want this process to actually occur sequentially, and we want cells to break apart into little clusters. What would be one way of trying to do this? Any thoughts? The first question, do we still activate everything at once, or what would be a workaround that? Right. So if there are no suggestions on this, so what we're going to try is sort of start off with the same epithelial layer that we have. Yeah, sorry, I can repeat the question. So the question is, suppose in the first problem that we explored, we began off with an epithelial tissue layer, and we wanted to create these groups of cells which have different properties at the rest. And so we selected out a particular region. Now, suppose we have a problem in which we want to sort of break the tissue apart in multiple groups. What would be some ways to do that? Suppose we still start off with the same epithelium, but instead of breaking them into a single cluster, we want to break our tissue into multiple clusters. And we could either do them all at the same time, or we could do them sequentially. Like what would be one way, what would be some of your thoughts on how we can achieve this? And we would be using FPP links again. So what we're going to actually do, and this code is available in module _5_1_3. I would suggest strongly that you download it. We'll walk over the code, but because this involves a lot of Python, it would be best if we walked over what the code is, and then we can work on the simulation and looking at the simulation output together. So the idea here is, if we want to sort of break the cell apart into different sort of clusters, one possible way is that we activate cells one at a time, starting sequentially from, say, the left of the simulation to the right, so that they develop their properties over time. And then, depending on how these developments induce secondary changes in their cell shape, we can use this as a metric to sort of like break apart the cell clusters. You could also sort of just activate, you sort of like increase the lambda all at once to everything else. And if you set the target distance so that they're only allowed a fixed target distance, you will see that the whole tissue will actually break down in chunks of like unequal lengths. But in the sequential case, if we are able to control the properties of the tissue as it proceeds from, say, the left to the right of the simulation here, we'll be able to produce clusters that are of uniform size, and we will also be able to mimic a sort of sequentiality. So we'll be able to get one cluster emerging before the other. Why is that something of interest? So back in grad school, Julia and I were working on this project of somatogenesis, which is basically trying to understand how a continuous epithelium periodically gives rise to distinct clusters. And one of the ways, by looking at these cell shapes from experimental data, what we saw was that these cells had a very constricted apical site, just like in the ones that we're simulating right now. And we thought that apical constriction could be a way to basically segregate this tissue into different clusters. So what we're basically trying to do is create up the epithelium, like we have done with the previous simulations, change the FPP properties either sequentially or simultaneously. And I'm hoping that you tried the simultaneous case a little while ago. But now we're going to try them sequentially and sort of see if we can get clusters that are uniformly shaped during the simulation. I'm just reading a chat message from Daria. Okay. So yeah, for CC3 related help, I can like circle back around in a minute, but I guess I'll let James answer that. Okay, so what are the changes to the code that we're going to be making? And the goal of this would be to look at the final simulation and plotting things that we can interpret. So for something that produces sequentially, what we're going to actually monitor, and I don't have the whole code in here. That's why I strongly suggest that you have module _3 open in front of you while we're going over this. So the idea is that we're going to sequentially start activating cells from the left-hand side of the simulation. So we're going to use two attributes. One of them is called the constrict attribute, and one of them is called the graph attribute. And these are ways of CompuCell creating dictionary objects that allows us to put certain attributes or properties associated with these cells. So our goal is, we want to start from the left side of the simulation. We want to slowly start increasing or changing FPP properties sequentially from the left to the right. But we want to know at what times, at what positions are these properties going to be changed. I'm going to pause for a second to look at the chat. I was wondering why you can't divide into intervals and invaginate. Why wouldn't that make? Right. So if you just divide the X domain into intervals, that's something that you are sort of forcing on the system, which is, I mean, it's a perfectly reasonable way to do it from the toy simulations. You can definitely say that, yes, these are my domains. Change your properties only within those. You might have some problems at the boundaries where cells will not know which of these edges to actually link to or form. And that would be probably give you where the boundaries are. But that would be some sort of an artificial case because you have superimposed the locations at which you expect your domains to form. The other way could be where we actually let the simulation evolve. The only things we're changing are maybe some the FPP properties, which could indicate levels of like actin massing locally at those regions, and let the simulation evolve to show us where it would like to form those breaks. So what the chunk of this code is trying to do is sort of going to is trying to pick up these is trying to find out what cell that we need to activate or start constricting. And then we're going to set an attribute of 1 to those cells that we want to constrict. We're going to make a sweeping wave from the left to the right. And we're going to do this at specific time points. So in the Monte Carlo step, what we're doing is we have some attribute of time. We have some attribute that tells us how often we'd like these cells to be activated. And every time we cross that amount of time, we're going to turn the constriction levels on. And we can also alternatively keep track of where that wave is at any moment in time. So we could basically use the attribute called graph here, which basically marks the position at which position along the x-axis of how many cells have been passed by this wave. So so what we're trying to do here is make a wave that goes from the left to the right, and only cells that have been passed by that wave, we're going to separate them from the rest of the simulation. We're going to set the constriction properties of those cells which have been passed by the wave as on, which is equal, we have set it to 1. And then we have another attribute called graph in the simulation that basically keeps track of where it is nearing the position. Is there a version of CompuCell that people are having the issue with that we could check, Hayden?
Yes, that would be better questions for James and Joel, I believe, right now.
Okay.
I'm not familiar with this particular problem.
Mm-hmm.
Like James says too, if it's just crashing without an error message, that would be useful for us to know as well.
Makes sense.
So we'll try to take care of that in the chat, Priyom. You can
Okay. All
go
right.
ahead and continue for now.
Thanks. So the other thing, so once we have identified the cells that we want to actually constrict, what we're going to do is we're going to now parse through all of our list of apical compartments that we have picked out for this process. So we're basically parsing out through the apical list in line number 75. We're going to each of these attributes which have the constrict level on. And if we have a neighboring cell, which also has the level of constriction on, we're going to actually change their FPP properties locally. And in this simulation, again, like we did with the simultaneous case, we could change either lambda distance or target distance or both. In this case, I'm choosing to keep the target distance the same as what we started off with when I created the simulation. And I'm only incrementing the lambda distance in like 0.05 intervals. And I also want to make sure that I am only constricting cells which have been if my right neighbor has also turned on constriction, that's when I start increasing any of the lambda properties. So I have two attributes, one identifying whether each cell has that constriction label on or not, and then making sure that whatever needs to constrict is to the right of this. So I wonder if it's a Python, sorry, I'm just looking at the chat message again, and I wonder if it's a version specific issue. So the codes were written with version 4.3. So if you're trying to run it on NanoHab, it might crash.
They should be running in 4.3 on NanoHub as well.
Oh, okay, awesome.
There are certain types of errors, like not closing a bracket, or if you comment a comment or embed a plugin inside another plugin by accident, that will cause CompuCell to crash without an error message. So if anybody's still having trouble with that, Pedro is in his breakout room to help with some of those issues.
Awesome, thank you. And James says that it shouldn't be version specific either, so that's good. Okay, awesome. All right.
Yeah, these are probably typos that are hard to catch. So like we said, Pedro can help in his breakout room.
I made sure they run before I uploaded them, so I'm not sure what they could be. Okay. So I'll just repeat what. Yeah, can you answer, James, that is it, are the simulations working for anyone, or are they crashing for everyone?
And if it is working for you, did you download it from the email that I sent out or from the Google Drive?
So my simulations were on my home computer, so I did not download them again. Okay, so Elena says it's running for me. I downloaded from the Google Drive. It's working for Norma. And for Lily.
Okay, so we have at least one confirmation from both download sources. Okay, yeah, so it isn't strictly the code. So it sounds like people who are having with I think people who are having problems are probably in Pedro's breakout room, so I don't want to spend too much more time talking to the group if it's still working. But if you are still having problems, it sounds like there's probably minor typos that Pedro can help you address.
Okay, thanks so much, Hayden.
So yeah, let's, in the interest of time, let's just go ahead and move on with the rest of the exercise.
Yep. So I'll just recap what we were talking about. So the idea is still that we want to sort of sequentially activate all of these cells. We are in the last couple of lines from the last slide, what we were trying to do is activate cells at specific times. And we were using the dictionary attribute of constrict and graph to sort of mark the cells that have been passed by that wave. Once we have collected the cells that have been passed by that wave using the attribute of constrict, as in line number 76, we went ahead and changed the FPP properties of these specific cell groups. So we did two things. We also made sure that if I am a cell which has the attribute of constricting, my cell to the right also has the same attribute, so that we could, once the cell to the right has been correctly attributed to have constrict, I start changing my FPP properties. And that's what I'm basically doing in line number 86 to 87, where once I've identified those specific links, I'm changing the lambda properties. And I am, once I've set once I have incremented my lambda value, I am setting back that lambda distance in the FPP link properties again. So these attributes of FPP links are like super helpful. So you can get the properties of FPP cells between any cell types. We can get the lambda distance that's currently in the simulation using something like get lambda distance. And if you want to adjust that value, like I'm doing in the next two lines, I'm also able to set back that value using set lambda. Okay. So once this has been done, we want to now sort of implement some sort of breaks in the tissue to form these little clusters that I was showing you a few slides ago. One way to sort of like do this is by looking at the simulation of a sequential like passing wave and realizing that as you have, suppose you're a cell to the extreme like boundary, you have more space to sort of maneuver and try to match the target distance. But if you're a cell in the middle, you don't have a lot of room to sort of like wriggle around and meet that required target distance. You can try like increasing the length of that apical compartment and try to like minimize that width, but you don't have a lot of choice. So if we look at these cell shapes and look at the elongated cells in the middle and realize that they're really miserable right now, and because they can't meet their target distance properly, and we set some sort of a parameter, which is basically trying to find out how far from that target distance they are actually at. And then say that if you are too far away from your target distance, and we can even we can sort of try to identify with a quantity that you might be familiar with, like tension. And we say that if the tension value has exceeded beyond a certain value, we would break that link. And that's what we're going to do now. So we first step we did was sequentially activate all of these cells. Next thing we did was we identified specific links which have both been activated by a constrict variable. We changed the properties of those links, and we realized that some cells are more miserable than others. And we're going to use that attribute to sort of break away or delete those FPP links so that we can get these isolated clusters. So this is where I have implemented the next step. So if you go back, and I don't have the line numbers on here, but it should be below the part where we just set that lambda distance. So now what we're going to do is we're going to get that target distance length, which is for each of these FPP lengths, what is that desired target length that they aim for. We're going to look at their actual length, and we're going to measure this quantity, which we're going to label as tension here. And we're going to see that how far are they actually from how far they have actually gone from their desired length. Note for each of these cells, we were also systematically increasing the lambda values, the lambda distance values. So in the final tension calculation, if you remember the original Hamiltonian that Julia showed you, that lambda value is also going to come into play. So the quantity that we're going to look at here is that tension, which is the lambda value for each of these cells. And we're going to multiply it with the amount of desired minus actual distance that the cell is able to adopt. And we're going to say, if you have a tension value, if you're in a much more tense state, here I've put a cutoff value of 400, and you're actually going to play with this value and tell me what happens. If you have a tension value of say 400 for this simulation, start breaking the links at those positions. And to actually break the link, we're going to be using the properties of delete FPP links. So what I want you to try right now is for the people for whom the simulation is working, which is 5_1_4, if you run it, you will see, maybe we can talk about what sort of behavior you actually see from the simulation. And we have a couple sort of like attributes. So we set up a speed at which each of these cells were getting activated. We had a measurement of the tension in this case, which is the lambda times the expected minus the target distance. And we were trying to match it off with a cutoff of 400. And so hopefully if you run the simulation, you should be able to see are you able to see clusters emerging? That's great. I'm glad it's working for people. So now you can sort of see that the clusters we created are sort of like uniform, mostly uniform. They contain like 4 to 5 cells in each. And we could also vary the length of the simulation, in this case, in the Python script, and sort of create more cells, and then basically have a better way to quantify the number of cells that form part of each cluster. The other thing that you could also play around with is are the parameters of tension, like we talked about. So we will be we also have a code that would let us like visualize how many cells form part of the cluster. But if the simulation is working for you, maybe what we can try now is change that tension cutoff to 250. So in the main steppables file, in the code that I uploaded, it's line number 92. So the tension threshold beyond which we have chosen the FPP links to sort of break. If you change it to 250 first, what do you observe as compared to the value of 400 that you just saw? I'm going to just wait a minute and let you tell me the answer. So that's basically going to line number 92, or the line that says if absolute tension greater than 400, we're replacing that 400 to 250 here. So basically setting it a lower level of tension. So saying that if even if the cells are stressed by 250 units, then cause the link to break. So maybe you can tell me what you see if you're running the simulation. Still seem a little loose. How do you mean? So more and smaller number of segments, absolutely. So now what's happening is that we have reduced that, reduced the threshold, and so that now these cells are able to break apart more easily than the rest. And if you actually compare the number of sort of the number of cells that make up a cluster, maybe you can compare whether you got more uniformity in the first case versus the other, or no. Perfect. So maybe we can also try increasing it up all the way to 500, and then, and maybe even 1,000, and you can tell me what you see. I'll again, like quickly like recap, because it might be a lot to take in. So we wanted to, we played around with a very uniform uniform constriction simulation where we chose all these cells to constrict all at once. But because we have the ability in Python to sort of manipulate the FPP properties over time, what we chose to do next was pick up cells from the left of the simulation to the right. We started changing them one at a time. And once we knew that neighbors on the right side were both sufficiently activated to be responsive to the constriction program, we started decreasing their lengths. And we kept doing this from the left to the right, and we saw that some cells seemed to behave differently than some others. And it was mostly because we were forcing them to really be constricted, but they had nowhere to go. I mean, the only way they could do is keep elongating their apical domain, but they had no other way of trying to meet that required target distance, which we were asking of them. And we could either do this by increasing the lambda FPP or the target FPP. In this case, we chose the lambda FPP. Once we did that, once we saw these changes, we asked them that if you're exceeding a certain threshold over which you're really miserable, why don't you just break that link apart? And we use FPP delete links to break that link. And once we did that, we saw that we got these clusters which form sequentially in time, even though we were incrementing lambda over from left to right. So a cell on the left would have had the highest level of lambda at any point than cells to the right. But what we saw was that we could get sequential clusters as they progress from left to right. Depending on the parameters that we chose, that level of like threshold of whether 250 versus 400 or 500, or I don't know if anybody's trying 1,000 and seeing if, I don't know if anybody tried 1,000, I'd be curious to know. So, but if you changed over those values, you could see that the clusters actually change. The number of cells actually changed in the cluster. In 250, you saw that cells were, there was more non-uniformity. You got maybe clusters of 3 cells at one point, or 2 cells, or even a single cell, which did not know which way to go. 750 led to one huge segment and one smaller one. Great. So see, so when you increase that level of threshold, it means that the cells are more they're more less sensitive to these, they can handle more of these tension levels. And so what you saw is they did not break, but once that first break occurred, you saw the resulting number of cells break off in the rest of the simulation. So that's great. I wonder if you even cranked it up, you could, if they would break it all, or if the simulation would do something weird altogether. But this is sort of a way in which, using just the tension threshold, you're able to control the size of the clusters. Now, one other thing we could try from here is we have, I don't know how much you have tried with scientific plots, experience with scientific plots at this point, but in version 5, version 1, version 3, without like looking at the, without actually looking at the result output, you could add a visualization plot, and you could try to just use the cell attribute graph that actually puts the position of where the breaks happen. That's what we updated in the previous slides, and you could actually try to plot the x positions of where these breaks occur. And now, if you know where these x positions are, if you subtract the if you subtract the positions of any two, you'll be able to find the number of cells that form part of the cluster. So your observation about whether something is uniform versus non-uniform, you can like sort of interpret from this. Okay, so I think Hayden's on, and it's time to for me to stop and conclude. So these simulations are available, and I can stay back to make sure that they are running. And if not, we can try to figure out something else. But try out the scientific plotting, try to find out the number of cells each of these clusters have. And if you have any questions, please let me know.
You can take about 5 more minutes if you need for Q&A and stuff, because the people in the breakout room are still finishing up too. So.
Okay, awesome. Yeah, I'll just let the slide, and then yeah, I'm happy to take questions. 1,000 lead to a float division by 0 error. That is an error I have not seen. I'm going to, after this model finishes, I'm going to try the values to see what could be causing the divide by 0. But yeah, if you have any other questions about creating compartmental links, one of the things that I did not have time to show today was how to create internal links as well. But you can basically switch on the internal tabs in the FPP plugins. And in the same way where we went to steppables, create FPP link, and then we created up links between different cells, we can also create those links between internal compartments. And we can do the same processes where we could change the lambda or the distance value between cells, sort of locate them. Okay. So I can guess what's going on. So in 5.1.4, there is, after the finish line, there is a part that basically lets you measure the number of cells that are in that cluster. So if the list is basically empty, and if there were no clusters that actually formed, it basically does not, there's nothing to divide it by. So if you look at 5.1.4, I had added an additional piece of code so that you have at the end of the simulation, if you wanted to find out on an average how many cells formed part of each segment, you will get a number at the end. So if at 1,000 there were no clustered form, the tissue did not segment at all because your tension threshold was so high, then you did not end up with any clusters. So if you want to fix that, you could say that only find the number of cells in the cluster if you have something in that list. But I can also update that in the exercise slides. Thank you for pointing that out. Yes, Megan. So what basically happens is that why we needed to sort of make that self counter.attribute is we have a bunch of cell IDs, right? And we wanted to make sure we collect only the apical compartments, and we wanted to make sure that they are labeled from the left to the right. Now, this list, the list of IDs, contains all the list of the important information of the apical members that I need. But there were 2 cells at the left and right of the simulation, which I had fixed and which was not what Julio had done. Now, the reason why I'd fixed them was, and why it's relevant to what Megan is asking, is the following. So in the periodic simulation, the links basically loop around because the right-hand side of your cell is actually connected to the left-hand side. But now if I wanted to impose some sort of sequentiality, I cannot let them be connected because otherwise they're going to rearrange their, the anterior or the leftmost cell is going to talk to the rightmost cell. So I needed to make sure that my 2 sides of the simulation does not talk to each other. So I put 2 very stiff cells with a very large lambda volume. And you can find this in the start function of the steppables in the Python file. Once I've done that, when I loop through all my apical numbers and I try to find out my list of apical cells, I will see that it's not just it's everybody, including those two very stiff cells. So that's why I had to subtract 2 to incriminate my counter. The other thing I had to do is, if you noticed in the code, what I was doing is every time as the wave was going from left to right, I was basically moving my counter of the apical cells that were being updated from left to right. So because I had no stop time to this, in principle, I could keep going on infinity and the counter would be updated every time that condition for sweeping the wave has been met. And I might run out of list members. So that line, that minimum of the counter value or the length of the list, lets me stop at the value so that my cell is not trying to find something with an ID that's insane, because I need only like reasonable IDs of the apical numbers. And if that counter loop runs till infinity, the simulation is going to give me an error of saying that this ID number does not exist. So I had to make sure I was within the right ID numbers. Hopefully that makes sense, Megan. If you have more questions on this, please let me know.